Have you ever tried to organize, I don't know, a messy bookshelf? It can be a pain, right? Well, imagine your job is to organize every single animal on planet Earth. We're talking about a puzzle with over 1 million pieces. Yep, that's not a typo. Scientists have described over 1 million different species of animals, from tiny worms you can't even see all the way up to the blue whale. The sheer variety is it's mind-boggling. A beautiful, chaotic library of life. So, how in the world do you start to make sense of all that? How do you figure out that a jellyfish and a jaguar, or a starfish and a spider, belong on different branches of the same family tree? Well, it turns out there's a system for it.
Okay, so let's get into it. Instead of getting bogged down in the tiny details of every single animal, biologists start by asking a few really big, fundamental questions. They look for a core blueprint, a set of architectural rules that tells them about an animal's basic body plan.
The very first question is all about complexity. How are its cells put together? Are they just a loose group of cells hanging out, kind of like in a sponge? That's what we call the cellular level. Or are those cells organized into proper tissues, which then form organs, and then full-blown organ systems, like what we have? Just asking this one question already draws a huge line separating the simplest animals from pretty much everyone else.
Next up on the checklist is symmetry. Now, this might sound simple, but it is a massive fork in the evolutionary road. It's really all about how you can slice up an animal and get two matching halves. And this breaks it down perfectly. You've got two main options. First, there's radial symmetry. Think of a jellyfish or a starfish. They're like a pizza. You can slice them pretty much any way through the center and get identical pieces. But then you have bilateral symmetry. That's us. That's an earthworm or an insect. We have a clear left side and a right side, and there is only one single plane you can cut through to get two mirror images. This simple difference tells you so much about how an animal moves and lives. All right, let's rewind all the way back to the very beginning of an animal's life, to the embryo. Another key design feature is how many layers of cells an embryo has. These so-called germ layers are basically the raw materials, the foundational stuff from which every tissue and organ in the body is built. So simpler animals, like jellyfish, are what we call diploblastic. They only have 2 layers, an outer one and an inner one. But almost every other animal, from a flatworm all the way up to us, is triploblastic. We have a third, middle layer called the mesoderm. And let me tell you, this extra layer is an absolute game changer. The mesoderm is what allows for the development of complex things like muscles, bones, and circulatory systems. And speaking of that all-important middle layer, the mesoderm, it makes another cool feature possible, an internal body cavity, also known as a coelom. This is the last big question in our blueprint. Now, this isn't just wasted space. It's a fluid-filled cavity that gives your organs room to grow and move around. It cushions them from shocks, and it can even act like a simple skeleton. And it turns out there are 3 possibilities here. An animal might have no cavity at all. That makes it an acolomate, like a flatworm. Or it might have a sort of false cavity, a pseudocoelom, which isn't completely lined by that mesoderm tissue. And finally, you have animals like us, the colomates. We have a true body cavity, totally surrounded by mesoderm, which allows for the most complex guts and organs. Okay, so we've got our 4 key architectural questions: organization, symmetry, germ layers, and the colum. Now let's see how biologists actually use this blueprint to build the family tree for the entire animal kingdom. It really becomes this very logical, step-by-step process, almost like a flowchart for life. You just ask these questions in order. First, what's the level of organization? Okay. Next, what's its symmetry? Got it. And finally, what's the deal with its body cavity? Each answer sends you down a different branch of the tree. And the major groups that come out of this whole process? Well, we call them phyla. Each phylum represents a fundamentally different and successful body plan that's been around for millions of years. Now, this is where it all comes together. Look at this chart and you can see the blueprint in action. You've got porifera, the sponges, way over on the simple end of things. Just cellular organization, no real symmetry, no colum. Then jump over to Annelida and Arthropoda. See? Both are complex with organ systems, bilateral symmetry, and a true colum. But they split off based on other key features like segmentation or having an exoskeleton. It's a system that creates order, but also really highlights the amazing diversity. And speaking of diversity, get a load of this fact.
That arthropod blueprint, the one with the exoskeleton and jointed legs, has been so unbelievably successful that it accounts for more than 2/3 of all known animal species on Earth. We're talking insects, spiders, crabs, the whole lot. Just wow.
So, with all this talk about other animals, you might be wondering, where do we fit in? Well, we belong to the phylum Chordata, so let's zoom in on our own branch of the tree and see what makes us who we are. Every single chordate, at some point in its life, has these 4 things in common. A notochord, which is this flexible support rod. A nerve cord that runs along the back, not the stomach. Gill slits in the throat area. And a tail that goes past the anus. And yes, that means you had all 4 of these when you were an embryo. And this slide really shows the massive difference. Our whole body plan is basically flipped upside down compared to a non-chordate like an insect. Our main nerve cord is on our back, theirs is on their belly. Our heart is on our front side, theirs is on their back. These are deep, fundamental differences in our architecture.
Okay, now here's a classic line that can kind of twist your brain at first, but it points to a really, really important distinction. So let's break it down. What's the difference between a chordate and a vertebrate?
And here it is. This is the key. Vertebrates are a special club within the chordates. We're the ones where that flexible notochord you have as an embryo gets replaced by a proper backbone, a vertebral column made of bone or cartilage. That one upgrade, that innovation, is what allowed for the evolution of big, complex animals like us.
So there you have it. From the simplest sponge to us, this simple blueprint brings order to over 1 million species. It shows you the deep history that's written into the body of every animal out there. So the next time you see a starfish on the beach, or a bug on the sidewalk, or even just look in the mirror, think about the symmetry, the layers, and the cavities that define its place and your own in the grand, spectacular tree of life.